So first a disclaimer, I will not be talking about images, but I will be talking about machine learning. So what I want to tell you about today is methods to select features in high dimension in the context when you don't have many samples. So I'm a researcher at Mines ParisTech and Institut Curie, which is, my office is really literally next door. So we work a lot on genomic data related to cancer problems. And my motivation here is something that is called precision medicine. So this is something that stems from the observation that because two patients present the same symptoms don't necessarily mean that they have the same disease in terms of how their disease is going to progress, so prognosis, or in terms of treatment. So they may not respond to the same treatment or respond in the same way. This is something you can observe in this. This is the first sentence on my slide. Sorry, it's behind me. There was a study in 2015 done on the top 10 highest grossing drugs in the US. So there's drugs given for fairly common indications, so cardiovascular diseases, diabetes, depression, things like that, asthma. And it turns out that those drugs, when they're first given, help between 1 out of 25 to 1 out of 4 patients. So this doesn't mean that we don't cure those patients, but it means they have to go back to their doctor and get a new treatment. So those differences between how people respond to drugs can be due to a number of things, including environmental, so what we call environmental variables, so whether you're a smoker or not, or clinical variables, so things like age or sex or other things like that. But we also know that differences in drug responses are partially due to genetic differences because of twin studies, in essence. And so the idea of precision medicine is, so not just about genetics, but that's what I'm working on, is to find ways to adapt treatment to the genetic specificities of the patient. And so there's an example of that, which is a drug that's called trastuzumab or Herceptin, which you give in certain cases of breast cancer. So when this gene, HER2, is overexpressed, so it means it produces more RNA normally. This dysregulation is part of the breast cancer phenomenon, and you can try to target it with this drug. If this gene is not overexpressed, it's not involved in the breast cancer of the patients, so even though you have the same symptom, which is to have a tumor in the breast. And in this case, giving this drug is not going to help the patient. So this is what we call a companion test. So there's a drug, but before administering it, you have to do a test. It can be a genetic test to decide whether or not it's going to help the patient. So what we would like to do is to go further in that direction and find genetic reasons to explain either why people develop a disease or how they respond to treatment in a much more systematic way. So in machine learning terms, the data we're working with is as follows. We have a number of measurements along the genome. So those are the little round things here. And we have also these genomes are genomes of samples, and those samples we have what we call a phenotype, which is an outcome if you want. So in this case I have a binary classification problem, so I have blue people and orange people, so cases and control. And I want to figure out which are the features, which are the regions in the genome, so the one in yellow here, that explain why some of my people are blue and some are orange. So one of the difficulties in this problem here is the size of the problem. So the number of features we're talking about are easily in the tens of thousands, hundreds of thousands, or even millions of variables, depending on which measurement you choose to use along the genome. Whereas you typically have a few orders of magnitude fewer samples. So this is the number of samples is not a problem that's going to go away with time in the sense that, first of all, we have, so I've talked about breast cancer, which is a fairly common disease. We're also studying sometimes very rare diseases, and if you work at Institut Curie and you see, or Redulme, and you see the children who come in for eye cancer, you don't want them to be more cases, to have more data to study the disease. And also what happens when you pull in more and more people is that they tend to come from more and more diverse genetic background, and this brings in some confounding, and sometimes it's better to have a smaller study on homogeneous population rather than having lots and lots of samples with, like, if you want, clusters inside that have different reasons for exhibiting the same symptoms. Okay, so we start with that situation. What I'm going to focus on for this talk, so sorry, we start with a situation, I didn't clarify this, where we have orders of magnitude samples fewer than dimensions, and so we are statistically in trouble. Okay, so what I want to focus on for this talk is something that's called genome-wide association studies, or GWAS, which is what I just described, where the genomic features are what we call SNPs, so for SNP, single nucleotide polymorphisms. So polymorphism means a mutation, and single nucleotide means it's a single letter of the DNA sequence, so you know, A, T, C, G. So in essence, we all more or less have the same DNA, that's why we're all humans, but not exactly, that's why we all look different. And one of the reasons one of those type of differences are those mutations. So we know along the genome places where the majority of the population has a certain allele, a certain letter, and there's a minority of the population which has the other one, and so that gives you a feature. Do you have the major or the minor letter in this position? And there's hundreds of thousands, actually tens of millions of those positions among the genomes, and so those are the features I'm going to work with. All right. So GWAS was a thing that came out like 15 years ago, and geneticists thought this was going to like solve everything. We had sequenced the genome, and thanks to this type of studies, we were going to be able to understand everything about our genomes. It turns out that the problem is much more complicated than that for many reasons, including the fact that we're not measuring everything that is involved in the problem, but also the fact that I've already mentioned that we're statistically underpowered because we have so many features and so few samples. And also because GWAS is traditionally conducted SNP by SNP. So you look at each of your variables and you ask whether it's associated with the outcome. So you can have like fancy statistical tests that take into account a number of things, but you're still looking at each SNP individually, each feature individually. So of course, there's a chance that you miss out on the joint effect of multiple SNP, multiple features. And that's the things I want to talk about today. Okay, so the first project I want to talk about is how to integrate prior knowledge in the studies to try to alleviate the two problems I've mentioned. And this is joint work. So this is work that started a while back when I was in Tübingen, so 6 years ago, and that is still ongoing with Hector Clemente, who's a PhD student with me at the moment. All right, so the idea here is that in biology, we're unlucky because we have so many features and so few samples. But we're lucky in the sense that we have a lot of prior knowledge that's been organized in a structured way. So there's an entire field in computational biology, which is called systems biology, which has, I mean, maybe they will find, people who are systems biologists will find this a caricature. But from my point of view, what they're doing is organizing knowledge about genetics and biology in groups and networks of things that work together. And this is very useful because we can use this as prior constraints on our problems. So the groups that you can have is genes that work together, so they're called pathways. But you can have also networks over your genes, over your region of the genomes. You can have different types of networks. They encode different types of information about the relationship between the genes, but the idea here is that you're going to be able to build a network where the nodes are features and the edges indicate on some sort of relationship between the features, and you have a strong belief that features that work toward the same outcome tend to be interconnected in that network. Okay, so this is what I call network-guided biomarker discovery. So biomarker discovery is my way, when I talk to geneticists, to talk about feature selection, which is a problem we're working on. So we have this biological network. It's given. It's over my features. So remember, hundreds of thousands, if not millions, or even 10 million of features, big network. With lots of nodes, which are my features. The edges encode relationship between those features, and I believe that my outcome is explained by features that are, so first of all, maybe I should say that I believe that my outcome is explained by a relatively small number of features. It's not the entire genome that is involved in the disease. It's only a few parts of the genome. And second of all, I believe that those features, they will tend to be interconnected on this network. So there's a number of ways you can do this, and if you're sitting in this room, you've probably heard about the lasso. And you may think that, well, the lasso is a way to give us a sparse solution, and maybe we can go one step further and add a constraint to also encode this network knowledge. So this is indeed something that was proposed in 2008 here. So you're, instead of just minimizing your square loss, so your empirical risk minimization, you add a regularization term. So if you stop here, this is a lasso. You enforce sparsity on your coefficient. And you can add a constraint here. That is a connectivity constraint. So it's based on the graph Laplacian. So you have your network. So W here is the adjacency matrix of the network you're using. The graph Laplacian can be defined in the ways it's here. And when you use this graph Laplacian here, what this means is that the weights beta are going to vary smoothly across the network. So if you have two nodes that are connected, they're going to have weights that are close to each other. And of course, here I've been using a linear model, which is an important point that I will come back to later. All right, so that's one way to do it. And remember, we are working with hundreds of thousands, if not millions of features, so P here is very large. Another way to do it is so something that's more directly inspired from, I mean, it looks a lot like the lasso and the network constraint lasso, but it's more directly inspired from the statistical tests that people are doing in this field of GWAS. It's what I called regularized relevance here. So the idea is that instead of looking for weights in a linear combination of the features, I'm just going to look at whether features should be selected or not. So I move from having a problem where I want to find weights in R to the P to a problem where I want to find a binary answer, 0 or 1, still 0 or 1 to the P. Okay, so to do this, instead of minimizing an empirical risk, I'm trying to maximize what I call here a relevance score, which is this function R here, which associates a score to a set of features. And this quantifies the importance of a subset of features for the outcome. So I can build this from statistical tests if they're additive, so meaning if you can build a test in such a way that the test statistic for the association of a group of variables is the sum of the test statistics for the individual variables in that group, which is fairly common. Here I have a caveat, which is that it's not because your test statistic is additive that the p-values are additive. So you may get a bigger test statistic, but not have a smaller p-value. But we work with what we can. Okay, so that's my first element. And then it's really, then it's also a regularization problem. I'm trying to find a set of variables that maximize my relevance. And I add a penalty that enforces my constraints about the problem. So I use a regularizer. So for instance, if I use for the regularizer the number of elements in my set of selected features, which is here, I have something that is equivalent to the lasso. And it turns out that I can also build a regularizer that is very similar to the one used to enforce this smoothness of the coefficient over the network in the lasso case. I can also build this in this setting. It's this term here. So if you look at it closely, so W is my adjacency matrix. And in essence, what this says, this is a number that increases whenever you have an edge of your network where one of the nodes is selected and the other is not selected. So if you have a lot of such edges, it means your solution is very disconnected and you have a strong penalty. Okay, and then you have this maximization of your score under the constraint that there should be few variables selected, and this connectivity constraint that they should be connected on the network. Okay, so the nice thing about this formulation, so as I said, we moved from trying to find weights in RP and then considering as selected the feature which had a non-zero weight, to a binary problem where we're in binary space. We just want to know whether a feature is involved or not. And so the nice thing about this formulation is that you can easily move from this equation here to what's called a minimum cut problem, which is something that people in computer vision are usually familiar with. So you take this is in black here, you have the networks that I was given initially. And you just need a slight modification of this network. So you need to add a source node and a sink node. You need to multiply the weights here. So the age weights, you need to multiply them by lambda, which is your regularization parameter, well, one of them. And you have a way to transform. So this is the score of a node. And this is the regularization parameter that's in front of the sparsity term. And so you can connect nodes from the source. So sorry, you can connect the source to one of your features or your features to the sink, depending on the sign of this term. And then it's a min-cut problem. And the nodes that are connected to the source in the end are the ones that are selected by this algorithm. So what min-cuts provides here is an easy way or fast way to solve this optimization problem exactly. So that's nice because it allows you to go much faster than the lasso version of the problem. Okay, so this is something we call scones because we were working on SNPs, so selecting connected explanatory SNPs. Sorry. I'm going to go quickly over the results because there's something else I want to talk about, and this is work that's starting to be a bit old, so some people in this room have heard me talk about it before. Okay, so we compared this thing to, well, what you would expect, so the network constraint lassos that I've presented before. Some univariate tests where you test the variable one by one, so mimicking what people typically do in this context. Lasso, where you don't have the network constraint. And when you have the network constraint, there's a variant of the network constraint lasso you can do, which is the overlapping group lasso, where you considered, so we considered either, we found a way two ways to build groups from our network to apply the overlapping group lasso. So the first graph I'm showing here is, it's simulated on a non-realistic network, and I should probably update this at some point to show it on a more realistic biological network. But what you see here is this red line, so this is the runtime, and this red line here is the runtime of our method with an increasing number of features. We have 1 million here, so we tested until 500,000 because that was the size of our data set back then. So it's much faster than the network constraint lasso counterpart. So we have two versions of a network constraint lasso. There's one that's faster, at least in a small number of features, but that requires more memory, and it crashes at some point. We can't run it further because we ran out of memory. Okay, so this is 2013, so probably everything would be much faster and run out of memory much later nowadays because we're 6 years later, but the story still scales. Okay, so this is the first thing. It does run faster, and then I have a number of simulations that show that so if you simulate your data in such a way that the causal features are connected on a network, our method is the one that's the best at recovering them. I mean, I could show you plots, but that would be the message. One thing that was interesting is that it was better than the network-constrained lasso. And I think this is because instead of looking for actual weights, you're just looking at whether those weights should be 0 or not. And we also checked that it was fairly robust to missing edges. So if you remove edges at random in your network, because you're not strongly enforcing that your solution should be a connected subnetwork, is just you're penalizing it if it's not. You're penalizing the solution if it's not well connected, but you're not enforcing that this should be one network, which is what a lot of methods in bioinformatics do when on this problem. It's more robust to missing edges, and we are expecting missing edges because I've told you we were lucky to have all this knowledge organized in networks and groups, but it's fairly, I mean, it's messy. It's noisy knowledge, it's evolving all the time, there's information that's missing, there's information that's wrong, so you can't rely too much on your networks. And of course it fails if the network is random, so if you feed it, if you enforce that your features should be connecting on the network and this network has nothing to do with the reality of the problem, then you have a worse solution than if you had not given any network. I think that's fair enough. Okay. We also looked at how predictive the features we had selected were. And what we found out so this is on real data, so it's on plant data. So we're a bit far from my precision medicine example, but the mechanics are exactly the same. So we had plant data for which we don't have ground truth, which is always a problem when you're trying to evaluate a feature selection problem. But in this case, we were studying phenotypes that were linked to growth of Arabidopsis thaliana. So Arabidopsis thaliana is a plant model organism, so it's been extensively studied. And one of the things that people who study plants are the most interested in is how they grow, because this is very important for agriculture and other applications. So we have good knowledge of which are the genes involved in growth, and so then we were trying to see whether we were recovering those genes or not. So it's shaky ground truth, interesting nonetheless. So we figure out that our method was selecting as many features as the other selected, so network-guided approaches. So if you add this network-guided constraint, you will have more features than the lasso because you will tend to take your neighbors with you. But they were tagging more candidate genes. So this is an indication that maybe it has more power to capture the actual phenomenon. And then we looked at whether those features we had selected were predicted of the phenotypes. And what we figured was that we had two cases. Either the lasso was outperforming all the network-constrained approaches, so indicating that the network is actually useless in this case, or not informative enough. And in the remaining cases, when one network-guided method was better than the lasso, then it was Cohn's, our method, that was outperforming the others. This is a line of work. So as I was saying, it's been published in 2013 for the first time. But we're still pursuing work in that direction. In particular, so the first thing we did, there was only one type of statistical test implemented. It was only for regression. You couldn't add covariates, so things like that. So now we've made it more usable in the sense that you have more statistical tests. You can also treat case control studies. You can incorporate covariates and also making it more usable for the target audience. It's now an R package on Bioconductor. So maybe this is not the right audience to promote an R package to, but if you're doing biostatistics or bioinformatics, this is what people are using. So it was important for us to have a bioconductor package. Okay, so that gives me some time to talk about another project in that line of work. Which is about interaction. So in the work I've presented so far, the effects of the variables on the outcome were additive. And there's no reason to believe that they should be only additive. And there's this like unicorn that people are chasing in Bino or in the GWAS community, which is this thing that we call epistasis, which is a joint effect between two features towards a phenotype and a non-additive effect. So there's different ways of defining epistasis. I didn't even write the word here. And what we've been looking at here, so together with Jean-Philippe, who's now at Google, and Lotfi and Clément Châtelain. So Lotfi is a PhD student. I'm co-supervising with Clément Châtelain, who's at Sanofi, so he's in the Thèse Cifre. So the problem we're looking at is the following. You have still your P variables. So I've hidden something from you before. I've told you that the features was you had either the major or the minor allele. So I kind of indicated that this was a binary variables we were looking at. But actually, you have DNA that comes from your mother and DNA that comes from your father. So you have two strands of DNA. So you can have either the major allele on both strands, in which case you have a 0. You can have the minor allele on both strands, in which case you have a 2. And you can have the major allele on one and the minor allele on the other, in which case you have a 1. So this is what we call a dosage encoding. The features get the value of how many of the minor version of the letter you have. So we have a target variable. So this one we've encoded in a binary way. So you have to figure out a way to go from this to this. So different encodings here give you different biological models. So in particular, if you think that only one copy of the mutation is enough, and it's called a dominant model. So having a 0 would be mapped to -1, and having 1 or 2 would be mapped to 1. Or you could have a recessive model in which you need to have both minor alleles to have an effect, in which case only the 2 would be mapped to 1 and 0 or 1 would be mapped to -1. And you have your outcome y, which for us here is also a binary variable, so it's queso control. And the question is, so we have this one target variable, so we have this SNP, we know, we like it, and we want to find if there's, so among the rest of the genomes, which are the variables that interact with this one SNP. So the reason we're asking this question is, so there's two reasons. Why is that? So one is that it's not really realistic to ask this question for all the SNPs in the genome, because remember, you have a million variables, and if you want to look at all pairwise effects between a million variables, then you're looking at 10 to the 12 pairwise effects. And if you didn't have the statistical power to find things in a million variables, you're certainly not going to find any signal in 10 to the 12 variables. So that's one thing. And the other is that if you have already identified that there's one mutation that has an effect on the phenotype you're interested in, well, then it's very rare that this, if you're looking at complex traits, this effect is going to be like, you're going to have some effect, but it's not going to explain all of the variation. So it's interesting to wonder whether this one feature, which already has a main effect, in combination maybe with other mutations in the genome, maybe this could explain more of the variation you see. Okay, so this is a problem we're asking. Okay, so we're not the first to ask this question. So there's actually, I was talking about the exhaustive approach. There's tools for the existing approach that exist. So one of them is called GBoost. It's from 2010, 2011, something like that. So the idea is they do the exhaustive search in a simple way. So you look at all your pairs of variables. So in our case, we will not do an exhaustive search. We will look at all the pairs of variable xj, a. And you would compare a logistic regression on xj and a, and a logistic regression on xj and a and their product. And you would do a statistical test to know whether this model explains the outcome better than this one. Because it's a fairly simple problem, but you're asking it on a number of times because you have all those xg variables, it's amenable to parallelization, and that's what the G in GBoost is about, is because it's implemented in GPU. Another thing you can do, if you're a fan of the lasso, is you say, okay, I'm going to do a lasso on all the variables that are involved in my system. So my target variables, my P other variables, and the cross products. So I'm running here a lasso. I would be running a lasso of the order of 2 P variables. And then I will see if some of those, the coefficients corresponding to those cross products come out as non-zero in my lasso. So we tried a different approach. And what we saw is you can model this outcome as a main effect coming from those P variables plus a cross product between the target variable and those P variables and some noise. And then if you can model this function delta in a sparse way, which I've talked about the lasso already, then the SNPs you're looking for, the ones that have a joint effect with A, are the ones that are included in that function delta. Okay, so we're not the first people to look at this type of models, and in particular, this is something that people do in clinical trials. So they have exactly the same question, but with different words in it. So we're wondering which of our SNPs and X interact with our target SNP A towards explaining phenotype Y. And in clinical trials, people are wondering which of the clinical covariates in X interact with the treatment A towards the outcome Y, so whether or not the treatment works. There's an interesting approach that was published in 2014 by Tian et al., which uses this exact model to answer the question. And they have a nice trick, which is called the modified outcome. They figured out that if they replace y by y prime, which is easy to compute, then they can model delta here as 1/2 of the expectation of y prime given x. And then they can model this in this relationship between y prime and x in a sparse way and find the answer to that question. And what's nice here is that once you have done this transformation, you have no need to model the main effects mu. So you're only evaluating the crossed effects here. So you gain some statistical power because you're not using any of your data to estimate mu. So we thought we're going to do the same thing. And so we looked into the computations that drove them to this modified outcome and to this equation. So that's where things got a bit complicated because if you write it out, so this comes from writing y as the expectation of y given a and x, and this can be decomposed in this product. If you look at it, if you stare at it long enough, you will see that mu and delta have to have those expressions. So first we had we realized we had to do a slight modification compared to them for everything to work out. So we had to modify our a. So a was encoded in -1, 1. We had to encode it on 0, 1. So this is all what this is doing. And then we arrived at this expression, and our modified outcome is this big thing. And in the case of the clinical trials, so A is the treatment and X are the clinical covariates, and because the same treatment has been given to all the patients in the study independently of, without looking at any of their clinical covariates, there is no dependence here. But in our case, which is what I wrote here, yes, in our case, you can't say that there is no dependence between one of the SNPs and the rest of the genome, because that's one of these things that is messy in those genome-wide association studies, is that there's a lot of correlations between the different features among the genome. And in particular, there's something that's called linkage disequilibrium. It means correlation, if you want. So the way cells are duplicated and reproduction occurs is that it's not like everything that is shuffled, not every position of the genome. It works by chunks. And so you have chunks of chromosomes that are inherited together, and all the variables on those chunks, they're very strongly correlated. And even then, even between those chunks, there are correlations. So you cannot say that there's independence between those variables. So unlike Tian et al., in the clinical trial setting, we cannot get rid of those terms. So we need to estimate them. So they're called propensity scores. So they're due to the structure of the genome, but this also means that we can leverage the structure of the genome to explain them, to model them. And so you can build a hidden Markov model where your hidden states are, so what in biology you'll call contiguous clusters of phased haplotypes. So those haplotypes are those blocks of correlated features, and phased mean estimated. So it means that we've clustered the features in blocks, and those clusters are our estimated haplotypes, and the emission states are the SNPs. So we haven't invented this model, and it's typically used for inputting missing values, and it's been used more recently. If you're familiar with knockoffs for false discovery rate controls, it's also what Candes et al. Have been using here. Okay, so we're almost ready to to compute our modified outcomes. We still have a little issue. Those propensity scores, they tend to be very close to 0 to 1. So you tend to have numbers here that are very close to 0. And numerically, this is not a very satisfactory situation. So you have several possible solutions. One is the computer scientist way. You have a number that is too close to 0, so I will add a small number here to all of those so that they are less close to 0 and that things are numerically stable. And then you have the statistician way, which I don't have time to explain, but you can use this robust modified outcome version where you have a good estimation of 1 over the propensity scores. And this problem is erased in this way. And so now what's left to do, so now you can model this term you're interested in. It's just a relationship between your SNPs and the modified outcome. So the modified outcome depends on the target SNP A. And you can model this relationship with an elastic net regression. So why elastic nets and not a lasso? It's because we have so many features and so few samples that the lasso is very, very unstable. So we're trying to stabilize it using the elastic net. So we will select more features, but if you run it several times, you will have more. It will be more often the same features that are selected. We still had to add stability selection on top of it. So this idea of stability selection is that you repeat, you do bootstrap samples, and then you rank the features according to how often they've been selected in those bootstrap samples. So we've been using a slightly different version of the bootstrap of the number of occurrences, which is one that was proposed by M. Claire-Horry in 2012. So you actually draw your regularization paths, and then you look at the area under the stability path. I don't really have time to go into the details. But it favors not only features that are selected often, but features that are entering the model early. Okay. And then you have, so the support of your solution here are the SNPs that you are selecting as being interacting with your target towards the outcome. So we did a number of simulations. In the interest of time, I'm presenting only one of them here. Those formulas are a bit ugly. What matters here is that, so we're doing a logistic regression, so a logit model of our case control phenotype. And we have SNPs that are synergetic with, so in epistasis with A, so the one we're looking for. We have some marginal effects, so some SNPs that are involved on their own in the outcome. And we have some quadratic effects between SNPs that are not the ones we're interested in. And we have 5,000 features, 500 samples, which is already pretty bad, and rather small effect sizes. So what you see here is that there's a slight improvement provided by our method, which is the blue line here. So there's a number of things on the slide that I haven't explained. The one I've explained here, so the robust modified outcome with the best estimation of this propensity scores is a blue line here. So you have here a ROC curve, here a precision recall curve. So this blue curve comes ahead. And for instance, GBoost is the magenta line here. So you see our precision recall curve is much better. And the ROC curve is also much better. Sorry, our ROC curve is also much better. And you also see, so some of those lines here are if you don't if you don't use corrections for those propensity scores, you see the results are worse. All right. So this is a tool we call EpiGWAS, so where we search for a sum of quadratic effects between our variables and our target variable of interest, while accounting for main effects, so effects of single variables, but without measuring them, without having to measure them, and modeling linkage disequilibrium. So this is so far a paper on bioRxiv and R package on CRAN. And this brings me to my conclusion. So there's a number of interesting questions still in this field. So one is the question of stability. So I've evoked it when I was talking about doing an elastic net and stability selection, but it's still not very stable in the end. So we have, and it's not very stable in the end, and it's very time consuming because you have an experiment that takes a lot of time and then you need to repeat it 1,000 times for stability, so that takes 1,000 times more. So there's lots of questions here. I've limited myself to either additive effects or quadratic effects. Things could be more complicated. There's one big thing I haven't talked about at all, which is statistical significance. So you can't really talk with biologists if you can't bring them p-values. So how significant is it that you have selected this or that variable in your model? So the approaches I've presented so far, well, as of yet, we don't know how to answer those questions. There's, however, a number of approaches that are emerging that are starting to allow to answer these questions on, on similar problems, different models. In particular, post-selection inference. So we're working at the moment on doing post-selection inference with using kernels to model complex interaction patterns. And another approach is to control FGR with knockoffs, which is something I mentioned before. And with this, I'd like to thank you for your attention on this rainy Tuesday morning. And so I have a number of people who were involved in this work to thank as well. And I'll take questions.
Thank you. Is there any question in the room?
Oh yes, while we're waiting for the question, it's a postdoc announcement things. So I don't have anyone who's looking for a postdoc, but I will offer a postdoc soon. I have money for it. I just need to put up an announcement. So if you're interested in working on this question, and you know someone who's looking for a postdoc and would be interested in working on this question that we're here, get in touch.
Thank you for your presentation. I missed the name of the R package that you showed. And
the first one or the second one?
Well, both of them. Would you be willing to show them again or write them down?
Sure. So for EpiGWAS, it's this one. So it's called EpiGWAS. And the other one, I won't go back to the slide. The other one is called Martini, like the drink. And you can you can find them if you go to my webpage. In any case, there's links towards them.
Other question?
Right, I think people want a coffee break. Even the mic wants a coffee break.
My mic doesn't work anymore. Just a small question.
Sure.
Is you said that the network is messy?
Yeah.
So do you have any random model for modeling for that, or?
No. For the way it's messy, no. So for how to build a network that is realistically like a biological network, yes. But I'm not really aware of any way of modeling how the information is missing or messy. I guess that would be good, because I've just been we're removing edges at random, but it's probably not missing at random, but I don't have any better model than that.
Okay, so thank you again.